So the point I was making here is that you have these long pathways, and it's easier to get a different result in a cell by altering a long pathway than by constructing a completely new pathway. And to maybe make some more sense out of that statement, think of an assembly line in a factory. If you imagine a factory that's making trucks, if you go out and buy a truck, you can buy a two-wheel drive truck or a four-wheel drive truck. It's the same basic truck, but I guess there's a slightly different transmission in one than the other. Well, I guarantee you they don't have two different factories making those two different types of trucks. They have one factory with one assembly line, and at the end of the assembly line, they probably have slightly different machines. One puts in the transmission for the two-wheel drive, and one puts in the transmission of the four-wheel drive. It's just more efficient to have one pathway and tweak it a little bit at the end to get a different result. Well, by having this long pathway like this, you can change some of keep the same receptor, the same G protein, the same first relay protein, the same second relay protein, the same third relay protein, and just have minor changes at the end to get a very different response from one cell to another. Let's see, yeah, there we go. So just a couple changes at the end gives you a very different cellular response, but the cell doesn't need a new different gene and new different protein for these beginning parts of the pathway. And the same with the lungs. The cell can use a lot of the same machinery, so to speak, a lot of the same relay proteins, but just have one or two different enzymes at the end of the pathway. So they think that's part of it. You can use a lot of the same proteins to get a different response in different cells. But another reason why they think there are so many steps in these cellular signal transduction pathways, this one I think is a stronger argument, is it allows amplification of the signals. And here's what that means. When I've been showing you these signal transduction pathways so far, I made it look as if there was, for each receptor, there was only one first relay protein, and there was only one second relay protein, and only one third relay protein, and one fourth, and one fifth, and one cellular response enzyme. And that was an oversimplification. The cell is actually loaded up with, I don't know, hundreds or thousands of G proteins and hundreds or thousands of the next relay protein and hundreds or even hundreds of thousands of the next relay protein, the next one, the next one. There's a lot of those different of those proteins floating around in the cytoplasm, and that allows for amplification of the signal. So imagine that here's a cell, and imagine that the cell only receives one signal molecule, just one epinephrine molecule has arrived. Well, that's going to bind to the receptor for it. So we've activated only one receptor. But when the G protein gets activated and then activates the adenylyl cyclase, that adenylyl cyclase is not going to make just one cyclic AMP, second messenger. It's going to make lots and lots because it's an enzyme. It can keep doing its reaction over and over again. So you might get hundreds of these cyclic AMPs, and each cyclic AMP can allosterically activate many of the relay protein, I guess this would be relay protein number 3. And each relay protein number 3, being an enzyme, can activate many, many, many relay protein number 4s. And those relay protein number 4s, being enzymes, can activate many, many, many of the cellular response enzyme. So by having so many steps, and with each step allowing for amplification, more molecules activated every step, you get, I don't know what you'd call it, an exponential increase in the response. You're going from one molecule arriving at the cell to millions and millions of cellular response enzymes being activated. You just get a very stronger, more robust response from even a small amount of signal molecule. And that's probably to me, that's the stronger argument, that the cell is more responsive to even small amounts of signal molecule if you have a big pathway with a lot of potential for amplification. You buy it?
It's kind of like in your car, when you have like a battery, small car battery, and you connect amp to it, you can run these like 20,000 or 1,000 watt speakers on just your car. You kind of find the signal.
I'll buy that. So you're one of those guys that drives around going.
I don't, but like it's kind of the same concept, right? So if you look at it from an engineering perspective, it's genius.
Okay, yeah, I agree. It's kind of amazing. Okay, so those are the two types of receptor we've talked about: the tyrosine kinase receptor family and the G protein linked receptor family, which is what all this was about. The last receptor type, signal transduction receptor type we're going to talk about are called ion channel receptors. Oops, go back. There we go. This type of signal molecule receptor, you're welcome. This type of signal molecule receptor is mostly found in nerve cells. It's these other ones that we've talked about, the tyrosine kinases and the G protein link receptors, you find in a lot of different cell types, but for the most part, the ion channel receptors are limited to nerve cells, which is what this yellow drawing is supposed to be. And to see one of these ion channel receptors in action, let's zoom in on the membrane of the neuron. Like so. And here's what they look like. Here is the binding site for the signal molecule. Now, incidentally, the signal molecules of that nerve cells use are called neurotransmitter molecules. But they're a type of paracrine. Remember that the paracrine was the term we used for signal molecules that just diffuse from one cell to another, that do not travel in the blood. Anyway, so the neurotransmitter molecules are just the paracrines that nerve cells use. Anyway, so there's the binding site for the neurotransmitter right there. And when the ion channel receptor binds the neurotransmitter paracrine, what happens is it opens up a transport area of the receptor to allow some ions to go inside the nerve cell. It opens up like that, and now you have an ion channel that can allow ions to flow inside the nerve cell. So this is kind of a neat protein if you think about it. It is a combination of receptor protein and transport protein. It definitely binds signal molecules, so it's a receptor-like protein, but it also can allow things to pass through the membrane, and that's a transport protein. So it's both. Now the one I'm showing you here, the neurotransmitter receptor protein here, is a sodium ion channel. It's letting a bunch of sodiums come inside the neuron. Some of them let calcium ions come into the neuron. Those are probably the two most common types of ion receptors, ion channel receptors. But there's certainly some other types. There's another type that lets chloride ions come into the cell. There's probably some other types that I'm not familiar with. But anyway, sodium and calcium are by far the most popular type. Now, so what's going to happen next when the sodium ions come in? You might think at first, based on what we've talked about, that these are going to be second messengers that are going to activate the next relay protein, which activates the next relay protein, that activates the next relay protein, until you get to a cellular response enzyme. But that turns out not to be the case. This is almost the end of the signal transduction pathway. What actually happens is this: when these positively charged ions come inside the neuron, that changes the voltage of the cell, because the positive sign of these ions means a positive electrical charge. And so as more positive electrical charge comes inside the cell becomes positively charged inside. Its voltage changes. And that is the response. That's the cellular response that the neuron was trying to get to. Because the job of neurons is to send messages between body parts. And just like this cable right here is sending messages between my computer and the projector by, I guess, voltage changes in the cable, that's what the neuron does. It changes its voltage as a way of sending messages between your brain and your muscles, or your sense organs in the brain. And so all it really has to do is change voltage. And here you see kind of a diagram of that. those little sparks going through her nerves, and within her nervous tissue, like her brain and her spinal cord, are the voltage changes in the neurons. And so that's all the cell really has to do. The neuron has to do is just let those ions in, and it's carried out its function. Sound all right? You're not disappointed that I don't have to go through 7 or 8 signal relay proteins to explain this one? Okay, well, maybe just as a closing thought, certainly nerve cells are a great example of where you find a lot of these ion channel receptors, but in actuality they're not strictly limited to nerve cells. Muscle cells have some also, though a smaller range of them, and I imagine you could find other examples of cells that have them, but certainly nerve cells are the best known. Okay, we are going to jump into a new topic now. Chromosomes. Let's see now. Just a couple of introductory remarks here. Up to this point in the semester, you've been learning a lot of biochemistry, which is usually meant to mean what the proteins in the cell are doing. And you've been learning some molecular biology also, which means what's the DNA and the RNA in the cell doing. From this point on in the semester, we're going to focus almost entirely on the DNA. You've had a pretty good introduction as to what the DNA is and how it functions, but now we're going to get into some more nitty-gritty details. As a matter of fact, I think there are around 11 weeks left in the semester. We're never really going to entirely leave DNA from here on in. It's going to be our main focus. And just to do a super quick recap of what we know about it so far, if this is the cell, and this is the nucleus of the cell, you remember that inside the nucleus are the chromosomes, which are big pieces of double-stranded DNA. And the DNA, what it is it encodes the instructions for protein. In the codons of the DNA are the recipes for which amino acids you link together to make the various proteins of the cell. But remember, the DNA itself doesn't come outside the nucleus to direct the assembly of the proteins. The DNA makes a what molecule? Messenger RNA, right? Which is a temporary disposable copy of one of the genes, each gene being a recipe for one protein. And then the messenger RNA carries that copy of the gene out into the cytoplasm, and who does the translating into the protein? What organelle? Ribosome. Right, there's this structure called a ribosome, organelle called a ribosome here, that with the help of the transfer RNAs brings amino acids inward to the ribosome, and reading the codons on the messenger RNA is able to assemble the protein. Let's say these are the amino acids strung together to make the protein. And then, proteins carry out tasks for the cell. And so now that may be an enzyme that changes the metabolic reactions in the cell, or it may be a receptor protein that the cell is going to use, or it may be a membrane transport protein that the cell is going to use. But in some way, this protein is going to contribute to the overall functioning of the cell, how the cell functions and what the functions of the cell are. This protein that's just been made is going to change the nature of the cell by giving the cell some new functions. And since you are made of your cells, that will give you some new traits. Maybe you think about a trait of yours, maybe if you have brown eyes, well, that's because the cells inside your eyes have some enzymes, some protein enzymes that make a brown pigment. If you grow to be a very tall person, it's probably because some of the cells in your bones have a protein that's extra active at making new bone. All of your physical traits come from the enzymes and other proteins that are in your cells. But those proteins themselves come from the information that are on your genes and your DNA. And so ultimately, it's really your DNA, your genes, that gives you your physical traits. They have to work by making the proteins, but these are like the workers. The DNA is like the architect who has the plans for making the initial building, so to speak. As I said, what we're going to be focusing on from here on in the semester is filling in more details in this process. And I want to start off by giving a little bit brief history of how this whole process was discovered. And for the most part, in this class, I haven't really emphasized too much the names of the people who discovered all these various things. I don't know, I was more partial just to explaining the facts behind something rather than who the discoverer was. But since we're going to be spending most of the semester talking about DNA, I thought at least learning a few names and what they discovered would be a good thing. So the story begins about 150 years ago with this gentleman right here. His name was Gregor Mendel, and he was a monk. He wore monk's robes and lived in a monastery in what used to be part of Austria, but the countries have changed a little bit now. I think it might now be in the Czech Republic. But in any event, he was the first person to discover the concept of genes. And this was before